Hello everyone, I again welcome you all on my YouTube channel Success Pharma. Today in this video, we will discuss about General Pharmacology Part 5. It includes enzyme. Enzyme is known as second target of many drugs. So, today we discuss only on enzymes. जैसे कि आपने लास्ट वीडियो में टाइप 3 एंड टाइप 4 रिसेप्टर्स देखे थे. Type 3 receptor is known as enzyme receptor. Some examples like cytokine, growth factors and insulin are come under the type 3 receptor, and some examples like steroidal drugs, vitamin A, vitamin D, thyroxine hormone are come under the type 4 receptors. So let's start the video. Now enzyme. Enzymes are the most important target for several drugs. Enzymes are the biocatalyst, optimize the rate of reactions and accelerated the rate of reactions. तो आपको 3 पॉइंट याद रखने हैं. It is known as a biocatalyst. Important is a biocatalyst and it optimized the rate of reactions and also accelerated the rate of reactions. So enzyme have three-dimensional structure of proteins. It consists of three-dimensional structure. So enzyme have three parts. First one is the apoenzymes. Apoenzyme, it is a proteinaceous part of enzyme. Enzyme without cofactor or coenzymes. Simply proteinaceous part means apoenzyme. Second one is the cofactor and coenzymes. The part of enzyme other than protein regulate the enzymatic activity. Other than proteinaceous part means cofactor and coenzymes. Again, cofactor and coenzymes are separate. Co-enzyme means if the part of enzyme is organic in nature, so it is called coenzymes. Example, NAD, NAP, biotin, ATP, FAD. ATP, adenosine triphosphate, etc. Second one is the cofactor. If the nature of that part is inorganic, it is called cofactor. Means एंजाइम का पार्ट जो अदर देन प्रोटीन है वो अगर इनऑर्गेनिक नेचर का होगा तो उसे हम कोफैक्टर बोलेंगे. इफ इट इज ऑर्गेनिक इन नेचर देन इट इज नोन एज कोएंजाइम्स. सो एग्जांपल ऑफ कोफैक्टर इज मैग्नीशियम आयन एंड कॉपर आयन एटसेट्रा. नेक्स्ट, होलोएंजाइम. इट इज अ कंप्लीट एंजाइम विथ apo-enzyme, coenzyme and cofactor. Holoenzyme means complete enzyme. Complete enzyme consists of apo-enzyme, cofactor and coenzymes. Next, steps of enzyme catalysis. तो जब भी एंजाइम की कैटालिसिस होती है वहां पे 3 स्टेप्स इन्वॉल्व होते हैं. फर्स्ट वन इज द substrate attached to the active site. Next, enzyme substrate complex, and next is the product formations. So first one is the formation of enzyme substrate complex. Second one is the generation of transition state complex, and last is the formation of reaction product, and finally free the enzymes. What is active site? Active site of an enzyme represents as the small region at which substrate bind and participate in the catalysis. Means active site enzyme की वह site होती है जहां पर substrate bind करके participate करता है catalysis और product formations. Now, silent features of active sites. The existence of active site is due to the tertiary structure of proteins. Means जो active site की existence है वो tertiary structure of protein के कारण है. Next, active site is made up of amino acids known as catalytic residue. Means active site हमेशा amino acids से बनी होती है. The substrate bind at the active site by weak non-covalent bonds. Non-covalent bond means weak interactions. Always the substrate bind to the active site by weak forces. Next, enzymes are specific in their function due to the existence of active site. So, the specific nature of enzyme is due to the presence of active site. The commonly found amino acids at active sites are serine, aspartate, histidine, cysteine, lysine, arginine, glutamate and tyrosine. Among these amino acids, Serine is the most frequently found amino acid. So serine is most useful. Next, the substrate bind to the enzyme at active site to form enzyme substrate complex. जब भी कैटालिसिस होता है तो सबसे पहले सबस्ट्रेट एंजाइम की एक्टिव साइट से बाइंड करेगा एंड एंजाइम सबस्ट्रेट कॉम्प्लेक्स फॉर्म होगा. नेक्स्ट, द प्रोडक्ट इज रिलीज आफ्टर द कैटालिसिस एंड द एंजाइम इज अवेलेबल फॉर रीयूज. और एंजाइम सबस्ट्रेट कॉम्प्लेक्स बनने के बाद फाइनली प्रोडक्ट फॉर्मेशन होगा और एंजाइम फ्री होगा फॉर द नेक्स्ट कैटालिसिस. नाउ enzyme substrate binding. So here blue color is for substrate and this substrate is bind to the active site of the enzyme and finally form products. This is the most important point in enzymes, that is theory of enzymes. So first one is the lock and key theory or Fisher's template theory. According to this theory, particular enzyme binds to the particular substrate on the basis of specific complementary shape. Means particular substrate ही particular enzyme से bind करेगा. हर एक substrate का अपना specific enzyme होगा. The active site has a rigid shape. Active site का shape rigid है. Only the substrate with the matching shape can fit and the substrate is the key that fit the lock of the active site of enzyme. Means यहाँ पर enzyme की active site act as a lock and key is the substrate. It fails to explain the stabilization of transition state and flexible nature of active site. इस थ्योरी ने active site को rigid बताया था जबकि active site flexible होती है. So it failed to explain the flexible nature of active site. It explained the specificity of enzyme substrate complex. So that is the lock and key model. Lock and key means lock is the enzyme, key is the substrate. When lock and key fit to form a enzyme substrate complex. Second one is the Salient model or Induced Fit Model. It is also known as modified lock and key theory. According to this theory, the active site of enzyme reshape continuously by binding to the substrate. Means active site का shape continuously change होगा after binding to the substrate. तो इस थ्योरी में हमें यह पता चला था कि एक्टिव साइट का शेप continuously change होता है. Next, according to this, enzyme are flexible in nature and active site are not rigid. तो एक्टिव साइट का शेप flexible होता है, it is not rigid. Next, induced fit model. Here E is for enzyme, S for the substrate. So active site is reshaped continuously and bind to the substrate and form enzyme substrate complex. Now, mechanism of enzyme catalysis. The formation of enzyme substrate complex, that is ES, is a very crucial for the catalysis to occur and for the product formations. So enzyme substrate complex formation is a very important step for all types of catalysis and product formation. Next, the enhancement in the rate of reaction is mainly due to 4 processes. तो जो catalysis की rate होगी उसे enhance करने के लिए 4 processes बहुत important हैं. First one is the acid base catalysis. Second, substrate train, covalent catalysis and entropy effects. Allosteric modifications. Allosteric site is the particular part of enzyme other than active site. It is very important. What is allosteric site? Allosteric site वह site होती है जो enzyme में another site होती है other than active site, in which the substrate bind and transfer the message to the active site and regulatory the enzymatic activity. Means वह साइट जहां पर सबस्ट्रेट बाइंड करेगा और कुछ मैसेज ट्रांसफर होंगे एक्टिव साइट पे एंड फाइनली एंजाइम परफॉर्म द रेगुलेटरी एक्टिविटीज. नेक्स्ट, काइनेटिक्स ऑफ एंजाइम. सो इट इज एक्सप्लेन्ड बाय Michaelis-Menten kinetics and follow the non-linear kinetics. So the equation is V equal to Vmax into S divided by S plus Km. S is the substrate concentration, Vmax is the maximum enzyme effect, V is the enzyme action or enzyme effects, and Km is the Michaelis-Menten constant. It is very much important for GPAT and NIPER exam. Thank you. प्लीज मेरे वीडियो को लाइक और सब्सक्राइब जरूर करें. एट लास्ट, काम करो ऐसे कि पहचान बन जाए, चलो ऐसे कि निशान बन जाए. अरे जिंदगी तो हर कोई काट लेता है, अगर दम है तो जियो ऐसे कि मिसाल बन जाए. थैंक यू सो मच.